Hi everyone. So today we will be discussing about DNA, its structure, different conformation, and its special arrangement, how it forms a supercoil. I hope it will be quite interesting, right? Okay. So without wasting a single minute, we will start with the basic structure of DNA. I hope that you have already uh, read, right, in your maybe 10+2. So if you see the DNA structure, we all know that DNA is kind of double helical, right? Kind of these things. But here we need to focus about the chemistry much. So DNA means the deoxyribonucleic acid, right? The first thing acid. Why it's called acid? Because of its nature, of course. So it has a phosphate group, right? And this phosphate group is acidic in nature. Okay, so that's why it's called DNA or deoxyribonucleic acid. Then why deoxy? That we will also see. See here in case of RNA, the structural unit. Okay, let's say this is the double helical structure, and this is made up of entirely sugar and phosphate backbone. Okay, so this sugar in case of RNA is the ribose sugar. If you see the ribose sugar, it's a ring structure here, right? This is number 1, this is number 2, number 3, number 4, and followed by number 5. So if you see the position number 2, in case of RNA, it's the hydroxyl group. But if you see in case of DNA, what it will be the normal age, deoxy. Okay, so that's why it's called deoxyribonucleic acid. In case of RNA, remember the 2' OH is there. In case of DNA, 2' OH is not there. That's why as in case of RNA, 2' OH is here. So RNA is less stable than DNA. Less stable. Why? Because this 2' OH is reactive. Okay, reactive means it can degrade the RNA. It can do some hydrolysis of the RNA, right? So the basic structure, the basic repetition is the sugar, phosphate, this thing only, nothing else. And then with sugar, some bases, nitrogen bases attached. And then this base forms some hydrogen bond or base stacking, that will come later. Okay, so in case of DNA, we have 4 different kind of nitrogenous base, A, T, G, C. A means adenine, thymine, guanine, cytosine. Okay, these all are deoxyribose sugar. That means they lack 2' OH. Okay, they have phosphate group. Because of this, it's acidic. You just write down the phosphate group is responsible for the acidic nature of the DNA. And the nucleotides are joined by 3,5 phosphodiester linkage. What is 3,5? I will tell you later. See, now focus here. Right. Yeah, you have to focus here. Okay, see, this is one ribose sugar, right? Here it's one ribose sugar, and in one ribose sugar, this is a 3' position, right? And in 3' position, one phosphate group is attached. This is the second ribose sugar, and this is the 5' position. If you see, I have did numbering here. That's the 5' position, right? In 5' position, OH was there. The OH is replaced by the phosphate group. Now, so these two sugars are connected by one phosphate group, right? That's why it's called phosphodiester. So it's making ester bond in both direction, right? That's why phosphodiester linkage 1,3 and 1,5. That's why 3,5 phosphodiester linkage. Okay. Now, see, the sugar is attached to the nitrogenous base in number 1 position. So this is the number 1, right? So we have to remember always the nitrogen base is attached at number 1 position. Okay. And now this bond is called glycosidic linkage. Okay. I'll write it down. This is called in glycosidic bond. Okay, this bond. Now we have to observe the structure of adenine and guanine. These are the purine bases. purine means they have two ring kind of structure, right? So, and pyrimidine means one base kind of structure. Okay, these are there are total 9 atoms inside the ring 10. Okay, now let's see. This is A, adenine. This is guanine. In case of adenine, the NH2 group is there. Okay, in case of guanine, both are there. So in here, one hydroxyl group, one NH2 group. So I hope you know the NH2 means N double H. O means simply O like this. So these are actually making hydrogen bond. That's what I need. You need to understand what is making, how the nucleotide, how the bases are making hydrogen bond because of this NH2OH, this kind of functional group, right? So always remember cytosine and thymine, they have the pyrimidine structure, and adenine and guanine are the purine structure. Okay, so and this is the number 9 position. So here the most important thing that you need to check, first thing is in case of RNA, hydroxyl group is present in 2' position. In case of DNA, it's absent. And later onwards, if you go with the 3 to 5 phosphodiester bond, so 2 sugar moieties are attached with 3,5 phosphodiester linkage. DNA is acidic because of the phosphate group. Okay, adenine and guanine, they are the purine bases. Cytosine and thymine, they are the pyrimidine. Okay, so purine pyrimidine, you have to just remember the structure a little bit, so it will be easier for you. You can easily draw the structure, you can make the hydrogen bond and all this. Next is the Watson-Crick model. So in our childhood also we have learned about this thing, that just 1 minute. Okay, so this is the double helical structure that Watson and Crick proposed. We know DNA is helical, but why helical? First question is, why helical? Why helical? This is the first question that should come in our mind, right? Second thing, we know that DNA just base pair like this. They have A pair with T, G pair with C. Why not A pair with G? Why not C pair with T? Right? We have to understand these kind of things, right? First thing, let's say the double-stranded helix model was proposed by Watson and Crick. That's why they got the Nobel Prize as well. The two feature is that to anti-parallel strand. Why anti-parallel strand? To anti-parallel strand wrap around each other. Let's say this is the 5' position, this is the 3' position. Again, this is 5', this is 3'. Why? Two strands are running exactly opposite to each other. Why so? Because if two strands are parallel, then hydrogen bonding will be very, very difficult. Okay, so just to make the molecule stable, actually the anti-parallel kind of rod is there. Okay, second question that I asked you, why G and C pair, why A and T pair, right? The thing is that here you have to see how much the base volume is. Base volume means whether the ring is bulky or not. Let's say you want to pair G and A, right? Then two bulky things will be there and it will not be stable. So one purine and one pyrimidine, right? That's how actually you can accommodate inside the DNA helical structure, right? And if let's say C and T are pairing, it's not I'm not saying that it's not possible. It can pair up, but the structure will not be stable. So just for providing the stability, right? In sake of stability, let's say one large thing, one small thing, right? This kind of things. So let's say this strand is carrying this large group, and this another anti-parallel strand is carrying this small group. So total volume is this much fixed. Now let's say if you have total right, these two only, these two, then what will happen? This much gap will be there, and again if similar to this kind of large groups are there, then they will also not be accommodated. So the packing should be like this, that the bases are accommodated properly and they are making some stability instead of making the molecule unstable. Okay, so that's why A always pair with T, G always pair with C. AT 2 hydrogen bond and GC 3 hydrogen bond. Okay, so remember, mostly the DNA is stabilized by few different kind of bonds that I will discuss later. But as of now, remember, hydrogen bond also plays a crucial role in stabilizing DNA. See, hydrogen bond is very weak bond. It's not very strong bond, right? But the thing is that if multiple hydrogen bond contribute, then the interaction will be very, very strong, right? Okay, so that's how if some GC repetition is there, that means this area is very, very stable. If some AT repetition is there, that indicate okay, this region may be some unstable. Okay, the diameter is 2 nanometer, or you can say 20 Armstrong. Diameter means this one. So this one is 2 nanometer. So you have to accommodate the bases in such a way so that they come in 2 nanometer. So it's not exactly straight line. See, bases are aligned like this. Okay. So distance between two pair is 0.34 nanometer. That means this distance is 3.4 Armstrong, and so from here to here, this distance is 34 Armstrong. Right? In one exact turn, 10 base pair, that approx 3.4 nanometer or 34 Armstrong. Okay, as we assuming for pDNA 10 base pair are there, that means the distance between, okay, gap between two bases is 34 divided by 10, that's exactly 3.4 Armstrong. Okay, sugar phosphate backbone outside and bases inside. So the sugar phosphate backbone means this is a sugar phosphate backbone, right? SP, and the bases are inside. Let's assume the bases are also outside. Then DNA will not be helical like this, right? If the bases are, let's say, this is the sugar phosphate backbone and the base is here, then DNA might not be helical, might not be bDNA, it's something different DNA, or some coiling kind of things will be there. Just think about this. This is quite beautiful to think, right? So you have to remember the structure of cytosine, guanine, adenine and uracil. There is no such tricky way, but first thing you just need to know how to draw the purine and pyrimidine. Nothing, it's very simple. Right, this is the purine and pyrimidine is the one, let's say here, yeah, this is the pyrimidine. It's quite simple. You just practice a little bit and it will be easy to you. Next one, the conformation of the DNA. Okay, DNA conformation. So conformation means DNA exists in different structural form. Here I am not talking about anti and syn. Okay. Okay, sorry. Yeah. Okay. So based on the situation, let's say different condition of solvent, salt, DNA can adopt different conformation. The most three studied one is the aDNA, bDNA, zDNA. Also CD, these forms are also there. Okay, so here we will discuss majorly these three things. First we will come to the aDNA. aDNA is right-handed helix. Okay, 11 base pair per turn, short and wide helix. Okay, deep, narrow, major group. I will discuss you the major and minor group. Okay, so when the aDNA is found mostly in dehydrated condition, and when the RNA DNA hybrid is there, it's some kind of, you can think of transcription format. In transcription, what happened? The DNA bubble is there, right, and RNA is also there. So here it's possible that the DNA can sometimes take the A conformation, right? So this is the RNA DNA hybrid. Okay. Next is the bDNA. This is a physiological form of DNA in living cell. This is right-handed. Okay, 10 base pair per turn, and in case of aDNA, remember it's 11 base pair per turn. Helical pitch is approx 3.4 nanometer. Okay, helical pitch means this one. This one is 3.4 nanometer or 34 Armstrong. I hope you know that 1 nanometer equals to 10 Armstrong, right? Okay, so base is perpendicular to the helix and standard Watson-Crick. So whatever Watson and Crick discussed, this is all about the BDNA. And when they consider the ZDNA, that's a left-handed helix. The only thing is very important that you need to remember, ZDNA is left-handed and 12 base pair per turn, and this is a zigzag sugar phosphate backbone, zigzag. Okay, and minor group, major group I will discuss later. So when ZDNA is formed, right? ZDNA is formed, it can be either GC-rich sequences. Let's say some of your DNA has higher amount of GC. In some condition, maybe in high salt condition, they can take Z conformation. Or in gene regulation role. Gene regulation means there is some ZDNA binding protein, and during some viral attack, something ZDNA plays some beautiful role. Okay, so best thing you have to remember that the high salt concentration actually promote ZDNA formation. Okay, so special thing about ZDNA, A and B DNA is the right-handed here, right-handed again here, but ZDNA is the left-handed. See, and ZDNA is zigzag phosphate backbone. And another thing is there unseen, but I don't want to bother you right now. Okay, good. Next, here is a basic comparison about ADNA, BDNA and ZDNA. Okay, same thing. Just give me 1 minute. Yeah. So this is helix type. First thing, ADNA is right-handed, BDNA is right-handed, ZDNA is left-handed. BP per turn. BP per turn means this thing, right? From here to here. How many base pair are there? In case of BDNA, it's 10 base pair. Okay. In case of A, it's 11. In case of Z, it's 12. So basically B, okay, sorry, B, A, and then Z, it's 12. Actually, it's dimeric form. So the diameter 2.3 nanometer, that means 23 Armstrong, 20 Armstrong, 18 Armstrong. So remember ADNA, it's wider. Here also you can see in the schematic, this is ADNA, it's quite wider, BDNA and ZDNA. The ZDNA structure, if you see, is a zigzag format. Zigzag format. Okay. Now group. So this is the deep major, clear major and minor, and narrow minor. So group means here you can see some groups are there. So while it's twisting, so the DNA is bending in such a way so that it, let's say, transcription or replication, this thing is happening. That means some protein is binding to DNA. DNA protein interaction. So this group actually plays beautiful role in DNA protein interaction. Okay. Just 1 minute. Yeah. See, here I have mentioned about the groups. Okay. So DNA has majorly two groups. One is the major group. So that major group is the protein binding side. Protein binding means here all the DNA polymerase, DNA pol, during replication, all the RNA pol, right? All different kind of transcription factor, different kind of repairing mechanism, everything can bind here in the major group. Here, how they will interact? Mean DNA and protein can either interact with the hydrogen bond between protein and SP backbone, sugar phosphate backbone, right? Or some DNA binding protein are there, those actually interact directly to the nitrogen base. Nitrogen base. That's a DNA protein interaction. DNA protein interaction. Minor group, these are the important for transcription factor recognition. Okay, minor group, so these are quite narrow. See the minor group, it's very narrow. See the major group, it's very deep here, and minor group is narrow. That's the difference between major group and minor group. So this group actually plays a beautiful role in gene regulation as well as some DNA protein interaction. The DNA protein interaction is why required? Because DNA polymerase, that's also protein, RNA polymerase, that's also protein. Those need to bind, right? Okay, next. Now one term comes, that's a DNA, denaturation and renaturation. As I told you, right, just focus on the figure as of now. It's a native double-stranded DNA. Now you just hate it. Then what will happen? At certain point of time, some portion will be single-stranded, some portion will be double-stranded, right? Partially unknown DNA. Then again heat. Then what will happen? These two strands will be separated, right? So red and blue, these are separated. Now this is single-stranded DNA, ssDNA. Now if you cool it, then what will happen again? They will make again the double-stranded DNA. This is called the renaturation. So from double-stranded DNA, right, dsDNA to ssDNA, that's called denaturation, and from ssDNA to again dsDNA, that's called the renaturation. How first the renaturation or denaturation will be, the kinetics mainly depends on the base composition. If GC is there, then as GC make 3 hydrogen bond, what will happen? They will renature first. If some repetitive sequence is there, it will renature first. So denaturation, the separation of strand by either pH. So let's say you have made change the pH to acidic or basic. Then what will happen? The hydrogen bonding pattern will be different, because some NH2 will be protonated, right? So because of this difference, the strand will be separated because lack of hydrogen bonding. Breaking a hydrogen bond, that's what it's saying. The denaturation is hydrogen bond breaking. It's not the sugar phosphate bond breaking. Don't consider like this. So once these two strands are separated, what will happen? Now the nitrogenous base are exposed. So as the nitrogenous base is exposed, that means it will absorb more UV light. See, here the bases are inside. They are properly packed. That's why the absorbance, UV absorbance will be less. Here the bases are exposed, right? So here it's exposed. As it's exposed, that's why the absorption will be more. So if you see the plot here, okay, see the plot here. Here it is single-stranded DNA, and the absorption. So this side is indicating the absorption of 260 nanometer, and that's the temperature. See, at 90-degree temperature, maximum of them are in single-stranded form, and because of this, the absorbance is much more. This is the partial thing. This is the double-stranded thing. The double-stranded thing is showing very less absorbance in comparison to the single-stranded. And let's say if you have just break the sugar phosphate backbone as well, and let's say it can happen for the free nucleotide, even the absorbance will be much more. Okay, so if someone asks you what is the absorbance rate, so the dsDNA is very less than the ssDNA, followed by free nucleotide. Okay, so renaturation, association of strand on cooling, nothing else. So just coming back to the native form of the DNA, and this increase in UV absorbance is known as hyperchromic shift or hyperchromic effect. Hyperchromic effect is nothing that so denaturation to renaturation, right? So when denaturation is happening, the strands are getting separated, and the bases, nitrogen bases, are getting more exposure. They are absorbing more, right? So there will be some absorbance difference between dsDNA and ssDNA. That's known as hyperchromic effect. Okay, cool. Then which forces actually stabilize DNA? See, whatever I am covering here, it's very much important in terms of exam point of view. I can cover in details, but that's not required for especially GAT-B, right? So what's the forces that actually stabilize the DNA? First thing is the hydrogen bonding that I already discussed, that between G and C, 3 hydrogen bonding, between A and T, 2 hydrogen bonding. This hydrogen bond also carries some energy, right? They will contribute. Another most important thing is the base stacking. Okay, and that's the most important thing. Base stacking actually contribute more stability than the hydrogen bond. So base stacking means let's say one base, another base. They have some overlap, some aromatic ring overlap, right? So aromatic cloud overlap, so electron cloud. So actually this base stacking contribute more. and followed up some hydrophobic interaction, some van der Waals interaction. But two major interaction is base stacking first, second is hydrogen bonding. Always remember, base stacking contribute more. Base stacking means how two bases. So base stacking basically is nothing, just the pi-pi interaction between aromatic ring of pyrimidine and purine. Okay, that's pretty simple and straightforward. Next, that's very important concept to understand. Supercoiling of DNA. See, supercoiling of DNA means DNA, let's say we have lot of genes, right? But our cell is very small. Our cell is this much, but our DNA is this much. Just as an assumption. Then how we can accommodate this inside this small dot? What we can do? We have to coil the DNA, right? Again coiling. So it's extreme coiling, and then only we can fit this particular DNA inside the small nucleus of the cell. That's known as supercoiling. Supercoiling means, let's say you have one particular wire. Let's say you have this wire. Okay, so this is the DNA. Now what you can do, first you just turn around this, okay, like this, and second you just have multiple round of coiling, maximum, just to accommodate. Now it's like this. That's known as supercoiling. So supercoiling actually involves two things. Okay, first thing is the overwinding or unwinding of the DNA. I will tell like overwinding and unwinding, or it can be positive or negative supercoil controlled by topoisomerase, important for DNA packaging. By this time you should understand that it's important for DNA packaging, right? That's what I told you, that you have to accommodate this whole gene, whole information inside some short space. How you can do this? That's a supercoiling procedure. Okay, so overwinding and unwinding. Let's see here how it's working. Let's say you have one circular DNA. Now, let's say, or maybe an easier way. Okay. Let's say 1, 2, 3, 4, 10. Okay. Let's say as of now you have total 10 and 10, 20 base pair. Now you have 2 turns. Now if someone tell you that you have to accommodate inside the 20 base pair inside one turn, then what will it will be? 20 base pair inside a single turn, or maybe let's say, yeah. So this is the 20 base pair. That's how you have accommodated, right? That means you are reducing the number of turn. But the base pair is same. But you are reducing the number of turn. That is known as the underwinding. And overwinding means, let's say 20 base pair is there, but you are giving 4 turn. So right now 2 turn is there. Okay. So 2 turn, 20 base pair. So if you do 4 turn, and in each turn, okay, 5 base pair will be there, and either you can say 1 turn. In each turn, 20 base pair will be there, right? So this is called 1 turn, means that's the unwinding. 4 turn means that's the overwinding. That means you are patching up and up. Now positive and negative. So this is actually controlled by topoisomerase. So let's say when DNA replication is happening, let's say when I will discuss the replication fork, I will tell you. See, when the fork is moving, okay, this direction. Now what will happen? In this particular region, it will be heavily, highly supercoiled form. Okay, let's say you are trying to different one rope, right? And you are just stretching it out. What will happen? In the later part of the rope, it will be too much coiling. So that's actually removed by topoisomerase. Okay, topoisomerase remove the positive supercoiling. Now that's for your imagination, like how actually supercoiling looks like. This is a double-stranded DNA. I'm just zooming it out. 1 minute. Sorry. Okay. See here. So this is the double-stranded DNA. Okay. The double-stranded DNA they are now packing up with the histone. So histone is the positively charged residue protein. So this is rich in lysine and arginine, K and R rich. So lysine and arginine means they're positively charged amino acid mostly, right? So they will interact with the negative charge of phosphate backbone, negative charge, and then they will neutralize the DNA, they will compact the DNA. Okay, so this is called nucleosome. There are 4 different kind of histones are there. Okay, I am writing here H1, H2A, H2B, H3, H4. 4 different kind of histones are there. So this is these 4 into 2. That means 2 copy of H2A, 2 copy of H2B, 2 copy of H3, 2 copy of H4. They actually form some nucleosome octamer. Okay, his octamer is known as the core nucleosome, and this core nucleosome is connected with H1. Okay, let's say one core, core nucleosome connected with other nucleosome through H1. So this is the H1. Okay, I hope it's clear. So remember, this histone protein actually helps in packaging. Okay, nothing else. So once it's packaged, okay, so it just makes a bead-on string kind of structure, and then through this again compaction and compaction, it just makes the chromatin fiber and finally chromosome. So in what is chromosome? Chromosome is nothing but the compacted version of DNA, and this compaction is happening because of the histone, because the histone has positively charged amino acid, right? Okay, so now we will focus about the linking number. Yeah. So what is linking number? See, linking number is a topological. Give me 1 minute. Linking number is a topological property. Topological property means you can't say it's a hard and fast rule. See, but property of circular DNA that describe how many times one DNA strand wraps around the other. See here, let's say here you have total 20 turns, total 20 twists. Okay, 20 twists is there. Linking number you can write down into formula. Linking number LK is equal to twisting plus writhing. Writhe means, here you can easily see, twisting means how many turns are there. This is called twisting. And let's say twisting, this kind of thing is happening, that's called writhing. So here you can easily say. So see here, L equals to 20, that means 20 turns are there, because T equals to 20. T means twisting was 20, and W, there is no writhing. So LK equals to 20, that is a twisting, plus writhing, that means 20. Now what happened here, 20 twists are there, but there is 2 writhing. Writhing means negatively, you are twisting in negative direction. So this is called negative supercoiling. So now you have changed W to -2, negative supercoiling. Okay, so because of this, right now the linking number will be 20 minus 2, that's the 80. So here actually the linking number is dropping. So how you can represent? You can represent through delta LK. Delta LK is equals to LK0 minus LK1. LK1 is the current linking number. Let's say here it's 18 minus 20, that's actually -2. Okay, and then another thing is just sigma. What sigma is nothing but delta LK divided by LK. That means -2 divided by 20, that's -0.1. That's represented here. Nothing is. And how it's triggered? It's triggered by DNA gyrase. So DNA gyrase introduce negative supercoilings sometimes. So it can change either T, twisting also it can change, or it can change writhing. But the thing is that in case of B DNA, how you can change the number of bases per turn? So mostly the W will be changed, not the T. Okay. So that's what it's mainly applied to covalently closed circular DNA. Please remember, covalently closed circular DNA, otherwise the concept will not exist. Linking number equals to twisting plus writhing. Twisting means TW, writhing means WR, right? And that's the summation. In case of negative supercoiling, W will be positively, that is a positive supercoiling. In case of negative supercoiling, it's a negative. Okay, and now delta LK means initial LK minus the current LK. So by seeing the linking number change, you can say whether it's positive supercoiling or whether it's a negative supercoiling. Okay. Okay. Next, go. Are you done? Yes. I hope we are done for today. Okay. So if you have any doubt, please let me know. Or we will discuss in next class about the DNA replication. It's also one beautiful topic as well.